Hello students, welcome to Sacred Heart High School. Today we are going to study in Science-2, Chapter 16, Heredity and Variation, Part 2. Before we proceed on to our topic, let's know about the scientist named Gregor Johann Mendel. Gregor Johann Mendel was an Austrian scientist. He studied the inheritance of some characteristics of the pea plant. He showed that inheritance of these characteristics follows certain principles. Later, this principle becomes popular by his name. Mendel's work was recognized only in the 20th century after a reconfirmation of these principles. The same principles now form the basis of modern genetics. So we can see that Gregor Johann Mendel was the first person who discovered the basic principles of heredity during the mid-19th century. Hence, he is known as the father of modern genetics. He observed a pattern of inheritance from one generation to another by conducting experiments on the pea plants in his garden. Genetic material is transferred in equal quantity from parents to progeny. As we had seen in Part 1, principles of heredity are based upon this fact. If both the parents make equal contribution to inheritance of characteristics, which characteristics will appear in the progeny? Mendel carried out research in this direction and put forth the principles of heredity responsible for such inheritance. The experiments performed by Mendel almost a century ago are quite astonishing. All the Mendel experiments were based upon the visible characteristics of the pea plant, that is, Pisum sativum. These characters are as follows, like shape of seed, color of flower, shape of pod, color of pod, position of flower, height of plant. So today we are going to study about the crosses to clearly understand the conclusions of Mendel's experiment. So we shall start with the first cross, that is monohybrid cross. Now why it's called monohybrid? In this experiment, Mendel brought about the cross between two pea plants with only one pair of contrasting characters. So hence this type of cross is called a monohybrid cross. So as to study the monohybrid ratio, let us consider the characteristics plant height with a pair of contrasting characteristics tall plant and dwarf plant. Tall pea plants and dwarf pea plant were used in this cross, hence this is parent generation, that is P1. Mendel referred to the tall and dwarf plant as dominant and recessive respectively. So, the tall plant was referred to as dominant because all the plants in the next generation were tall. The dwarf plant was referred to as recessive because these characteristics did not appear in next generation at all. Now, this experiment has been presented by the Punnett square method. So, as you can see in the picture that P1, there were two plants with two different characters were taken, tall and dwarf, but in F1 generation it was seen that all the four plants had shown the characters of tall. Now, depending upon these observations, Mendel proposed that factors responsible for inheritance of characteristics are present in pairs. Today we refer to these factors as genes. Dominant genes are denoted by capital letters, whereas recessive genes are denoted by small letters, as you can see in the Punnett square. As genes are present in pairs, tall plants are denoted by capital T and dwarf plants are denoted by paired small t. During gametogenesis, these genes separate from each other due to these two types of gametes, one containing factor for tall and the other containing factor for dwarf. Now, in this experiment, Mendel observed that all plants of first failure generation, that is F1 generation, were different from the tall plants of P1 generation. Why? Because parents of F1 tall plants are tall as well as dwarf plants. Depending upon observation of F1 generation, he concluded that factors of tall plants are dominant over the factors of dwarf plants. Though all the plants in F1 generation were tall, they contain the factors responsible for dwarfness. That is, though this phenotype of F1 plants is tall, their genotype is mixed. Now, what is phenotype? Phenotype means external appearance of visible characteristics of organism. For example, tallness or dwarfness of plant, whereas genotype means the pairs of genes responsible for the visible characteristics. Now, genotype of P1 tall plant is double G, as you can see, and produces only one type of gamete. Genotype of F1 tall plants is heterozygous and it produces two types of gametes. Thus, based on this, we can say that in case of tall plants of F1 and P1 generation, though they show similar phenotype, the genotypes are different. Mendel further continued this experiment and brought about self-fertilization in F1 plants, from which a second failure F2 generation was produced. Now you can see in the second generation, that is F2, both that is tall and dwarf type of plants appeared. According to the data collected by Mendel, out of 929 pea plants, 705 were tall and 224 were short. Thus, the phenotype ratio of this type is 3:1 and genotypic ratio is 1:2:1. So, it can be referred that in P2 generation, phenotypically there are two types of plants, whereas genotypically there are three types, as homozygous tall plants, homozygous dwarf plants, and heterozygous tall plants. Next, we shall see with the next type of cross called Mendel's Dihybrid cross. In Dihybrid cross, two pairs of contrasting characters are under consideration. Mendel performed more experiments on hybridization in which he considered more than one pair of contrasting characteristics. So we can say that Dihybrid cross is the cross between two different genes that differ in two observed traits. By monohybrid cross, only Mendel could first find the inheritance of one gene in the plant. So Mendel studied the subsequent 7 characters, that is stem height, tall dwarf, seed shape, round wrinkle, seed color, yellow, green, pod color, green, yellow, pod shape, inflated, constricted, flower color, violet, white, flower position, axial or terminal. So a hybridization is a breeding experiment between two organisms which are identical hybrids for two traits. In other words, a dihybrid cross is a cross between two organisms with both being heterozygous for two different traits. Now, the individuals in this type of trait are homozygous for a specific trait, and these traits are determined by DNA segments called genes. So, in a hybridization, the parents carry different pair of alleles for every trait. One parent carries homozygous dominant allele, while the other one carries homozygous recessive allele. So, the offspring produced after the crosses in the F1 generation are all heterozygous for specific traits. Now, Mendel took a pair of contrasting traits together for crossing. For example, he took color and the shape of the seeds at a time. He chose the green wrinkled seed and round yellow seed and crossed them. He obtained only round yellow seeds in the F1 generation. This indicated that round shape and yellow color of seeds are dominant in nature, while the wrinkled shape and green color of seeds are different traits. Then F1 progeny was self-pollinated. This resulted in 4 different combinations of seeds in the F2 generation. So they were wrinkled yellow, round yellow, wrinkled green seeds, and round green within the phenotypic ratio of 9:3:1. As you can see from the picture. So he considered capital Y for yellow color seed and small letter y for green color seed, capital R for round shaped seeds and small letter r for wrinkled seed shape. Thus the parental genotype can be concluded as yellow round seeds and green wrinkled seeds. So it was found that F2 plants formed by fusion of 4 types of male gametes and 4 types of female gametes had phenotypes such as round yellow, round green and wrinkled green with a ratio of 9:3:3:1, whereas genotypic ratio as 1:2:1:2:4 2:1:2:1. So hope children you have understood Mendel's dihybrid cross and monohybrid cross. Thank you children.